Okay, today I want to show off this really educational paper. It's titled Direct Evidence for Microbial Derived Soil Organic Matter Formation and its Ecophysiological Controls. So, what this paper is demonstrating is the true nature of soil organic matter. So, of course, we're all familiar with how important soil organic matter is, but its origins are a little elusive and historically have been presented as if all soil organic matter is derived from plants, and this paper points out that no, that's not exactly true. Soil organic matter accumulation is greatest in soils with higher fungal abundance and more efficient microbial biomass production. So, in this paper, we're going to dive in and see exactly why that's the case. So they start out with some context, and I'll just read and highlight as we go. For nearly a century, soil organic matter formation in conceptual and quantitative models has been depicted primarily as a function of plant inputs and their chemistry. As such, chemically diverse and stable soil organic matter originates from the preservation of biochemically recalcitrant complex plant polymers, such as lignin derivatives and long chain lipids. So, right off the bat, they're saying that for the past 100 years, we've basically believed that soil organic matter was accumulation from plant biomass, is what they're saying, is plants would grow and then they would eventually die and they would fall onto the soil. And what they say is there's recalcitrant complex plant polymers. So that just means there's parts of the plant's body that are really difficult to break down, such as the lignin and the long chain lipids. And that because they couldn't be broken down, this was the main constituent of soil organic matter. So next I go on. The importance of soil microbes in processing plant inputs and synthesizing soil organic matter is not conceptually new, though until recently has been largely overlooked and primary pathway of soil organic formation. Okay, so they're saying, yeah, the idea that microbes are the key to creating soil organic matter is not new, but hey, everyone's overlooking that it's actually not just the key, it's like the main way. It's almost like the only way this occurs. Of course, you do have plant biomass and you do have the roots of plants in the soil, but that is just a food source for the microbes. So the microbes, as we will see, are really the ones building soil organic matter. So here we go. Mounting evidence demonstrates that some decomposition-resistant soil organic matter bears little chemical resemblance to plant material, but is instead characteristic of microbial cells, excretions, and cytoplasmic material. So they're calling it out. They're saying, you know what? We've studied soil organic matter long enough, and it actually looks nothing like any of the plant polymers or derivatives from the plant tissue that we know of. Instead, it looks a lot like microbial cell excretions and the cytoplasm of these microbial cells. And just as a side note here, so they call out that these cytoplasmic materials can be stabilized via organo-mineral and organo-metal complex interactions. So that's just saying that if we're attributing the microbial cytoplasm and excretions to being the key source of soil organic matter, how is this soil organic matter actually stabilized? And that's kind of another topic, but they're alluding to the fact that it's interactions with the minerals in the soil, the clay, and other metals. So that's just a side note. Okay, so they go on to say that plant residues that accumulate in the soil through physical protection, i.e., inside aggregates or zones with low biological activity, are susceptible to destabilization following disturbances such as cultivation or in response to environmental change, example, temperature. If, however, plant materials are synthesized into microbial proteins, lipids, or polysaccharides, the resulting organomineral associations may include a lignin bond or other strong interactions that have a lower temperature sensitivity and may be better withstand perturbations. Despite the potential importance of microbial-derived soil organic matter, experimental evidence for microbial contributions to soil organic matter formation is constrained by methods that select for a limited group of microbial. Okay, blah, blah, blah. They're saying there's not a lot of evidence out there. It's hard to get evidence. But the thing to note in this passage here is that they're calling out that plant residues, if they are to be the source of soil organic matter, it's not very stable because you could till the soil and you could transfer the plant tissue from a location that's protected from microbes and stable to one that is not protected from microbes or stable, or the soil could warm up and microbes could then access the plant tissues and therefore they would consume these plant tissues and thus the soil organic matter isn't stable if that's the case. But they're suggesting that the plant materials are actually synthesized into microbial proteins, lipids, and polysaccharides, and that these become complexed in organomineral associations. Okay, so let's continue on here. So down here, they mentioned that as carbon use efficiency, that's what CUE stands for, increases, relatively more substrate carbon goes towards biomass synthesis, potentially increasing the amount of residues available for stabilization. Okay, so this is a very kind of important point here, is carbon use efficiency. So this is basically when microbiology has access to a carbon source, how much of that carbon source can it take in and utilize as part of its body and metabolic function versus how much does it respire away? Okay, so you have to think of carbon going into the microorganism and going out of the microorganism through respiration. Bacteria respire a lot of the carbon that they consume. Fungi do not. So fungi have a higher carbon use efficiency, and I hope that made some kind of sense. So we use model soils to quantitatively assess whether microbial processing of simple carbon substrates alone in the absence of complex plant compounds can build significant amount of chemically diverse stable soil organic matter. So we're slowly getting into the experiment that's about to take place here. But what they're saying is basically they're going to create these little mesocosms, little, piles of soil and put them in an incubator and feed them carbon, just simple carbon sources, so sugar, no plant derivatives, no lignin, nothing like that, nothing from a plant. And they call out that the sugars that they're putting in are abundant energy sources for microbial metabolism in natural soils. Okay, so why are they putting sugar onto their artificial soils is basically to replicate the root exudates of plants. So we'll go into that more later. But let's move on here. So again, they keep touching on this point here, and it actually is really important. Microbial soil organic matter accumulation being greatest in soils where fungal abundance are highest and microbial biomass production is most efficient. So what they're literally calling back to is this here. They're saying that, yeah, if you have microorganisms that have higher carbon use efficiency in your soil, such as fungi, yeah, you're going to be generating substantially more soil organic matter. And remember, they are saying here in this paper that soil organic matter is a product of microbes and their growth and their metabolism and their excretions. It has nothing to do with the plant biomass. So here we go into the most exciting part of this paper. So here's their soil mesocosm I mentioned. It's just a jar full of soil. It's an artificial soil. It's sterile. It's just a mixture of clay and sand. And it looks like dirt at day 0. Here it is, day 0, it looks like dirt. Now, over time, look, it starts becoming more aggregated. There's all these aggregates forming. And then you see a lot more biological activity on the surface, probably fungi or some sort of actinomycete or something like that. And then look, 15 months later, we went from this to this. I mean, that's amazing. Look at what this looks like. Really well aggregated, rich, fertile soil. I mean, that looks like good soil. And it only took 15 months. So remember, what happened is what the researchers did is they added simple carbon sources. So they did inoculate the soil, of course, and then they fed it sugar. And they went from this to this in 15 months. So here's the first figure that they have. All right. So they have a field soil. So this is their field soil. They sampled the field soil, and they actually extracted microbiology from this field soil and inoculated this dirt at day 0 here, okay? And then, like I said, they fed it sugar every week until at the end of 15 months, you get this. So this is their field soil, and what they're doing is they're quantifying the different chemical compounds in the soil. So proteins, lipids, chitin, nitrogen-bearing polysaccharides, phenolics, lignin derivatives, aromatics, and other. So they have all these classifications of compounds, and they sample a field soil, and they break down these classifications. So this is what a field soil looks like right there. Now, what they do, here you see sugar-treated soils. These are 2 different types of clay. That's it. It's just simple clay. So dirt. And at time 0, this is what their clay, this is what it looked like, okay? What is it composed of? Well, it's composed of mostly polysaccharides. But notice it looks nothing like a field soil. This, at day 0, looks nothing like a field soil. Now, after 18 months, both of these different clays that were treated with sugar, what do they look like? They look like a field soil. So they have every aspect of a field soil in almost the right ratios. I mean, look at how close this is. It's so close. I mean, they basically turned dirt into something that looked like a field soil, and they didn't add any plants, none. They just fed the biology. So this is extremely important to understand what's happening here. And then you'll notice that they have other figures here. And so this was the sugar-treated soils. And then we have Serengal-treated soils, which is another carbon source. And then this is dissolved organic carbon-treated soils, which is kind of just like a plant tea. It's just literally a tea, kind of. But the most important one is the sugar-treated soil, because this is actually replicating root exudates from plants. So look down here, they call out that chitin, let's see here. Chitin originally originated primarily from fungal cell walls, also increased by 5% after 18 months. So 5%. So what are they talking about? Look, here at day 0, where do you hardly see any chitin, if any. What is chitin? Chitin is this one. So maybe, I don't even know if that's chitin right there. But what happened is, yeah, there's now 5% more chitin. So it's looking, it's even outperforming the field soil in a way. So here's a crucial passage here. In contrast to traditional expectations that slowly decomposing plant compounds accumulate soil organic matter, lignin derivatives specific to plant-derived lignin macromolecules decline from 10 to 2.5%. Relative abundance of dissolved organic carbon-treated soils and protein and lipid increased fivefold. Okay, so let's break down what they're saying here, because this is amazing. Let's clean this up. Look, so they're saying in contrast to traditional expectations. Basically, traditional expectations would have it that we have lignin derivatives, which are specific to plant macromolecules. You would expect, if you were accumulating soil organic matter, and that accumulation could be contributed to the macromolecules that are contained within plant tissue, that those should increase. But no, they actually declined from 10 to 2.5%. So the very thing everyone used to think was driving the formation of soil organic matter is actually going down, while soil organic matter is going up. So that's a huge discrepancy from what would be traditionally expected. But what was going up, so relative abundance, proteins, lipids, and proteins and lipids increased fivefold. Okay, so again, we have the compounds associated with plant tissues going down, and the compounds associated with microbial residues going up, while soil organic matter is increasing. So what does that tell you? It tells you that what's driving soil organic matter formation are proteins, lipids, which are derived from the microbial bodies. Both after death and while the microbes are actively growing, they are excreting extracellular metabolites. Okay, so after seeing this photo, you might be wondering, wow, in 15 months, this went from looking like dirt to looking like soil. How much organic matter is in that final sample? And here it is. At the end of the carbon addition period, 1 to 1.4% over 15 months. That's incredible. I mean, traditionally, no one would accept that to be the case. That shouldn't really be possible, honestly. Most agricultural soils are within this range of 1 to 1.4%. So they took it from almost nothing to an agricultural soil in just over a year. So that's pretty amazing. And again, what did they add besides biology? They added some field soil biology extraction. But what did they add to feed the biology? Sugar. Simple sugar, which, remember, is supposed to represent the root exudates of green growing plants. Okay, now, if this wasn't fantastic enough, if this wasn't amazing enough, they, kind of put the cherry on top and they say the majority of the soil organic carbon was biologically stable and was chemically stable to oxidation, which is within the upper range of observed observations for natural soils. So, not only did they produce an incredible amount of soil organic matter using nothing but the activity of microbes, but that soil organic matter was actually found to be really stable. So, just like you would imagine if you were looking at a field soil. So, by all accounts, they've replicated a field soil. Alrighty, let's finish it off. So, they used real-time monitoring of microbial soil organic matter formation and demonstrated that microbial processing of simple carbon substrates, sugar, produced an abundance of stable, chemically diverse soil organic matter. Wow, it's amazing. And these were dominated by proteins and lipids. So here we have the protein, lipid, and chitin classes derived exclusively from microbes constituted 34% of the soil organic matter compound relative abundance across all samples. So they generated soil organic matter, but 34% of it could be attributed to the protein, the lipids, and the chitins produced by the microbes. Now, then they go on to say many of the other compounds not derived from lipids or proteins may also be derived from microbial residues. What do they mean by this? Essentially, the other components that were not accounted for by the protein, lipid, and chitin classes were probably extracellular metabolites from the actively growing microbes. So more complex polysaccharides that are responsible for gluing soil aggregates together. Oh yeah, here they say it, extracellular polysaccharides. So this is a very important one here. Extracellular polysaccharides are part of soil organic matter. So this is what glues your soil together. Okay, so let's just finish it off here and try to highlight the key learnings. And what I want to point out is that these researchers were able to build soil organic matter in a controlled setting simply by feeding the microorganisms in their model soils with sugar, simple sugar. Okay, and so how much simple sugar did they feed their microcosms? Well, they specify here 0.7 milligrams carbon per gram dry soil. So then they say the rate of weekly carbon additions reflects the upper range of natural carbon inputs to soil from daily root exudates. So yes, they are trying to replicate daily root exudates. And the important thing here is that they are giving this experiment the best possible conditions, so to speak, because they are maintaining the soil at a warm temperature via incubation, allowing for lots of microbial activity, and they are feeding the soil at a rate that would be well within nature's capability, but probably the upper bounds of what nature would be able to do. So this strong soil organic matter accumulation over 15 months, you probably would not be able to replicate this in reality, but maybe give it 2 years and you could. So it's certainly relevant and well within the range of what nature could produce. Now, they did also treat the soil with inorganic N. Oh, that hurts. So a nutrient solution, 1.56 milligrams N per gram soil. So Yeah, they used synthetic fertilizer in this experiment. They did put some nutrient, some inorganic nutrient onto the soil with their carbon additions. And you know what? Your soil, as much as this hurts, yeah, you can feed your soil inorganic N. Anything under probably 100 pounds per acre isn't going to wipe out your soil microbiology. In this experiment, it certainly did not wipe out the soil microbiology. It was able to continue on rapidly forming soil organic matter. Okay, I think that's the end of this paper. So let's just look at this beautiful uh, image one more time before we call it a day. Wow. So, this is what microbiology can do to your soil. This is the point, is that if you feed the microbiology in your soil, it will take it from looking like dirt to looking like absolute beautiful soil. Look at that. Okay, that's all.